So now we'll continue on with our story about antibodies. So again, work with the terminology here. Antigens, I always have a lot of students who wind up flip-flopping antigen and antibody. Antigens are molecules that your immune system responds to. Antibodies are one of the weapons that your immune system generates to help fight off infection. So we'll talk a little bit more about these antibody weapons. All right, so here we've got a diagram. This is kind of your generic, typical antibody diagram that you see in any class where you talk about the immune system. All right, so a typical antibody, it's a protein. So it's a sequence of amino acids, just like we learned about back in Chapter 8. And an antibody that looks like this, where you have the Y-shaped molecule, consists of 4 polypeptide chains, 4 proteins. So that gets into, remember, quaternary structure that some proteins have, where you actually have multiple polypeptides that get together and assemble to make a fully functional protein. So this is an example of that. And if you notice here, here's one polypeptide chain, here's another one, here's another one, and here's another one. So 2 of them are shorter chains, and 2 of them are longer chains. The longer ones are called heavy chains, and the shorter ones are called light chains, which makes sense based on their molecular mass. All right, then notice here in the reddish shaded colors, these are areas of antibodies that essentially are the same, okay, for any antibody. Those areas, those are called the constant regions, so they're going to look the same on any antibody molecule. Here at the tips of the antibody, though, this is where you have your variable region. Okay, so think about, we go back to B-cell clones. Any individual B-cell clone has receptors that attach to one particular antigen. And then if that B-cell becomes stimulated and starts generating plasma cells, those plasma cells are going to generate antibodies that attach to that very same antigen. So we've got to be able to produce many different kinds of antibodies that attach to essentially any antigen that we would get exposed to. So the basis for that diversity is right here at the tips. So these are called the variable regions, where you have a 3-dimensional pocket that attaches to a specific epitope of an antigen. Remember we said if my fist is an antigen, like my knuckle right here might be an epitope for an antibody to attach to. And then my pinky over here might be another epitope for other antibodies to attach to. Another name for that is antigenic determinant. Here's another look at this. So again, with antibodies, you've got light chains, 2 of those, 2 heavy chains, and then it's right here at the tip. This is where, that's the business portion, that's where binding to an actual antigen takes place. And here they're kind of blowing this up for you in this figure and showing you that you really do have a 3-dimensional shaped pocket here in the variable region portion of the antibody. And then here's the epitope, the actual section portion of the antigen that fits inside that pocket. And so this should be a very nice, very specific fit. So if this antibody, if this is some glycoprotein on the surface of influenza, this antibody is only going to essentially attach to that particular type of molecule. It's not going to attach to an antigen from another type of virus. It's not going to attach to one of your antigens, hopefully, and it's not going to attach to, say, a molecule on the surface of Staphylococcus aureus bacteria. It's very, very, very specific. Let's see, some other things. This portion of the antibody, the stem, is sometimes called the FC region. You'll see that mentioned in your textbook as well. Another thing I want to point out over here on this diagram from your textbook, there's a couple of things that I like. One, down here at the very bottom of the stem, or the FC portion of the antibody, binding site for cells. So there are receptors on your phagocytic cells that attach to antibodies. So that'll wind up being important in a little bit when we talk about antibody function. And then remember when we talked about the complement protein system at the end of Chapter 12? Notice there's a complement binding site on the stems of some antibodies. So when an antibody, in some cases, when some types of antibodies attach to a microorganism, that will attract in complement proteins that set off that whole complement cascade that we talked about. And then that can lead to the poor little microbe bursting, if you remember that from the end of chapter 12. That's sometimes called fixing complement, the antibodies that can do that. So all these things wind up working together to help you clear out infections. All right, so what do antibodies do? How do these things... Okay, so we know these are proteins, and they attach to very specific molecules on the surfaces of microbes or other things that are foreign to the body. So how in the world does that help us? Okay, so if we've got some sort of generic bacterial cell here, and this bacterial organism has stimulated a B cell response, and so we've got antibody molecules that are being secreted, and those leave the lymph node, spleen, wherever, and they wind up entering, they leave the blood, they go into the area of infection, and they can start attaching to the poor little microorganism. And since that microorganism is generally going to be covered with multiple copies of the antigen, you can see that more than one antibody can attach to the surface. Remember how we said when we talked about complement at the end of chapter 12, that C3b complement protein can opsonize microorganisms, certain types of microorganisms, which means they coat the surface. And this is called antibody opsonization, or coating of a microorganism as well. All right, we just mentioned over here that the stem portions of antibodies have binding sites for cells. So there are receptors on phagocytic cells that attach to the stems of antibodies. So this is one of the key things, this is one of the key ways that antibodies help clear out infectious organisms, because with all those stems sticking out from the surface, those can attach to receptors on phagocytic cells like macrophages, for example. And so that's going to help attract the phagocytic cell to the antibody-covered microorganism. And so now the poor little microorganism is going to get engulfed and shredded to pieces, and it's going to wind up dying. So thinking back to chapter 11, we talked about pathogenic bacteria that have things like capsules. You got Streptococcus pyogenes that has M proteins. We talked about how Mycobacterium tuberculosis, the mycolic acid cell wall, prevents it from being phagocytosed easily. Interestingly enough, once you activate this third line of defense, you're going to generate antibodies that attach to things like capsules, antibodies that attach to M proteins, antibodies that attach to the surface of Mycobacterium tuberculosis. So, and now these bacteria that weren't getting phagocytosed very well before because they have those protective coatings and so forth, once you make antibodies, all bets are off. Now the antibodies are like magnets for phagocytic cells, and those pathogens are going to get engulfed anyway. So basically, ultimately, our immune system wins the battle. The critical thing is just making sure you survive long enough for your antibody response to come in and help you win that battle. So antibodies coating a microorganism and attracting phagocytic cells, very, very important antibody function. And then notice what's going on down here. So a typical Y-shaped antibody has two binding sites. Okay, so each one can bind a separate copy of the same antigen. And what that does is that helps cross-link multiple copies of the same microorganism together. So if you have a bacterial infection somewhere in your body or a viral infection somewhere in your body, it's not like you're just going to have 3 or 4 copies of the bacterial cell or 3 or 4 viral particles. There are going to be gazillions of them in all likelihood. Once you start making antibodies, once antibodies start infiltrating in there and attaching to those microorganisms, because you do have 2 binding sites, that's going to help cross-link those together. You're going to be able to make clumps of these microorganisms. And so why is that an advantage? Think about your phagocytic cells over here, your macrophages and your neutrophils and so forth. If they have to find each individual microbe, that's not very efficient. But these phagocytic cells are pretty big, so imagine that they can engulf all at the same time, 100 bacterial cells or 200 viral particles, because these antibodies have clumped them all together. So that winds up making phagocytosis more efficient. You can get rid of more of those all at the same time. That is called agglutination. Let me write that down. Of course, that term is in your textbook, but I'll go ahead and write that down now. Agglutination. You'll hear about that when you do the blood typing part of your Learn Smart Labs activity on the blood with this particular unit. Agglutination is a term for clumping. If you've done blood typing before, maybe you did that in Biology 202, you no doubt talked about agglutination, the clumping of the red blood cells that helps you identify your blood type when you do that particular test. All right, so what else can antibodies do? Notice over here we've got some generic virus particles, and antibodies have attached to the surface of the virus particle. That allows antibodies to neutralize these viruses. And what does that mean? It doesn't have anything to do with pH. Remember that viruses have to attach to host cell receptors. If the virus is covered with antibodies, that binding is going to be blocked. So basically neutralization here means the virus is blocked from attaching to its host cell receptor. And same thing for other microbes as well. If you coat bacterial cells with antibodies, and let's say they have fimbriae on their surface that have to attach to a particular tissue, that's going to be blocked. That's going to be neutralized. Remember the toxins we talked about. Remember all those nasty, evil bacterial exotoxins we learned about in Chapter 11. You can generate antibodies to those toxins, and now the toxins can be neutralized. The toxins can't go into the cells that they're supposed to harm and cause damage. So you can have neutralization of toxins as well. That's an important concept with antibodies to remember. And that's what's shown down here. Antibodies that attach to toxins are sometimes called antitoxins, or like a snake venom or a spider venom. Maybe you've heard of that called as an antivenom or an antivenin. Or like if you watch some movie and we've got to come up with the antidote. It's kind of that concept. So if you were bitten by a snake and exposed to the venom of a poisonous snake, you're going to generate antibodies that will attach to that snake venom. So those are called antivenom or antivenin molecules. But those are antibodies that help you recover from something like that. So toxins, venoms, viruses can be neutralized. Other types of microbes can be neutralized, and that's through blocking. They can't go where they're supposed to be able to go. And then finally, that complement system. So at the end of Chapter 12, we talked about how antibodies can attract in the complement proteins and set off that cascade that causes many different kinds of microorganisms to wind up bursting or rupturing. And there are viruses that can get damaged by complement as well. So antibodies attaching first is one of the things that can activate complement. All right, another concept with antibodies is they don't all look alike. There are different classes of antibodies that can have somewhat different functions and locations where they carry out their business. All right, so one concept that's kind of important, not kind of important, it is important. If a B cell gets stimulated, goes through all that rigmarole we talked about in the last video lecture, goes on to make plasma cells, so the clones expand out, they convert into plasma cells. The plasma cells start secreting antibodies. Those plasma cells will go through phases where they secrete different classes of antibodies. So the tips, the variable regions of these different antibody classes, they're going to attach to the same antigenic determinant, the same epitope, but the antibodies themselves have some different features and characteristics that provide different functions. So there's some tables in your textbook that review characteristics of these different antibody classes. So kind of that generic Y-shaped antibody that we always talk about, that one is a class of antibody known as IgG. So Ig stands for immunoglobulin. That's another name for an antibody. Antibodies have kind of the light chains and the heavy chains have a globule, a globby type shape. So immunoglobulins are antibody proteins. That's abbreviated as Ig. And then kind of the generic Y-shaped antibody is in this IgG class. And you'll hear this type of terminology used. So you want to be at least a little bit familiar with it. And this table's pretty nice. It shows us how many antigen binding sites. One right there, one right there. So that's going to be 2. So IgG antibodies could bind to 2 separate copies of the same antigen. Molecular weight is given there, 150,000. What percentage of the total antibodies in your serum, that's your blood fluid, 80%. Half-life in serum. So antibodies don't last forever. So once a plasma cell starts secreting IgG antibodies, an individual molecule on average is going to be able to remain in your serum and stay functional for about 23 days. Anybody know what the difference between plasma and serum is? That's a good concept to go over, because we've talked about how plasma is the fluid in your blood. Serum is fluid in clotted blood, so the clotting factors have been removed. So that's your difference between serum and plasma. You know, for our purposes, you can generally think about those 2 terms meaning the same thing. That's the fluid portion of your blood. Look at this, crosses the placenta. Can IgG antibodies cross the placenta from mother to developing embryo or fetus? Yes. Fixes complement. Can it activate that complement system? Yes. Does the stem portion over here attach to phagocytes? Yes. What are the biological functions? This is called an antibody monomer as well, because you just have a single Y-shaped molecule. This is your most prevalent type of antibody, circulating in your tissue fluids. So they get out of the blood, they move into your tissue fluids. And the functions here are the ones we just talked about: neutralizing toxins, opsonizing, fixing complement. If you opsonize, that's going to attract in the little stem portion here. The FC portion attaches to phagocytes. So if you got a microbe covered with these IgG antibodies, those IgG antibodies are going to help attract in the phagocytes for phagocytosis. So these have got lots of interesting things going on. Now how about this type? Let's take a look at this one next. IgM class of antibodies. This is a pentamer of the Y-shaped antibody. So you actually have 5 of those connected together. And there are some other features here in the center that help hold all 5 copies of those together. So one IgM can attach to 10 copies of the same antigen molecule. So those are going to be really good at clumping, that agglutination we talked about, because a single antibody could attach to 10 different copies of the same microorganism. So 10 antigen binding sites. They don't last very long, though. They only have a half-life of about 5 days. Y'all know what half-life means? So if you had, let's say you had 100 trillion copies of the same antibody floating around in your bloodstream, after 5 days, you would be down to 50 trillion. After 5 more days, you'd be down to 25 trillion. After 5 more days, you'd be down to 12.5 trillion. So it's the amount of time that it takes for the concentration to drop by half. IgM antibodies are big, so notice they can't cross the placenta, so they're not going to help a developing embryo or fetus fight off any infections. They do activate the complement, fixes the complement system. And these are the first ones. So we said an activated plasma cell will go through phases where it makes different versions of antibodies that are attached to the same antigen. So these IgM ones are actually made first, which kind of makes sense if you think about it, because early in an infection is when you're going to have the most copies of a microorganism. So if these guys are really, really good at agglutination, early in an infection, it's good. You want to kind of mop up and agglutinate as many of those microorganisms as you can. Now let's take a look at this one. So IgA. This is a dimer. So you have two copies of the same antibody, two Y shapes. And they're held together in the center by something called the J chain. That's another protein that holds them together. And notice that little green squiggly right there? That's called the secretory component. So that helps these IgA antibodies remain stable and dissolved in mucus, basically in bodily secretion. So you tend to find these secreted onto mucous membrane surfaces, like in your respiratory tract, in your digestive tract, and elsewhere. Also, this is important. You've no doubt heard that mothers who breastfeed pass their antibodies on to the nursing infant. This is the class of antibody that gets passed from the mother to the nursing newborn. So that's something to keep in mind as well. And that secretory component there helps protect those antibodies from getting digested, getting broken apart by the infant's digestive system. Because these are proteins. So when you guys study the digestive system in Biology 202, you guys learn about how proteins get broken apart by your digestive system. You have digestive enzymes that chop those apart so you can use those amino acids for your own purposes. So that secretory component there is going to help protect those antibodies from getting destroyed by the infant's digestive system, and then they're able to get absorbed into the bloodstream. All right, the IgD type antibody here, that's the B cell receptor. So those are the versions of the antibodies that are actually sitting in the surface of a B cell and functioning as the B cell receptors that we've talked about. And then finally, IgE over here, that's another Y-shaped antibody type. Notice the stem's a little bit longer. Very short half-life. Doesn't cross the placenta, doesn't fix complement. And the stems on IgE attach to mast cells and basophils. We're going to hear about these more. We're going to hear about IgE-type antibodies more when we get to Chapter 14. Because these are antibodies that are involved in allergies. So if you are allergic to something like oak pollen or whatever, or some kind of pet dander or whatever, you are producing IgE-type antibodies against that antigen that's triggering your allergy. And it's an inappropriate production of these. You're producing those in a case where you're not really supposed to. IgE antibodies are important for worm infections, helminth infections. So that's probably one of their major roles. They're really supposed to be the antibodies that attach to parasitic worms and play a role then in attracting in the cells that are going to kill the worms. They're also involved in anaphylactic reactions, like if you go into anaphylactic shock, an extreme allergic reaction, and they play roles in asthma. So we'll hear more about those guys when we get into Chapter 14. All right, and so this slide is going over special properties of the IgA antibodies right here that we just talked about, and I think I covered all of this. Yeah, these are special antibodies that you secrete. Instead of being in your blood and your tissue fluids, you secrete those onto the surfaces of your mucous membranes, and that's great. Remember, your mucous membranes are portals of entry, correct? Those are barrier locations. So, hey, it's great that you secrete antibodies onto those surfaces to help mop up any microorganisms that you're fighting off that are living in those locations. And again, as I mentioned, IgA, and these are the only type of antibodies in colostrum. That's the early milk produced by a newly nursing mother, and they're not digested in the stomach or the small intestine of the infant. That's very important. Then they're actually absorbed through the lining of the infant's small intestine, and they get into the infant's bloodstream. So when you're a brand newborn baby, and you guys know this, your immune system is not fully functional yet. And so you need it's good for you to have some assistance by receiving antibodies from mama when you are breastfeeding. Because that makes sense when you think about it. If the mom is getting exposed to something and having an immune response to it, the baby's going to get exposed to the same thing. So it's great that the infant can tap into the mama's antibody response for a while. All right, now how about primary and secondary responses to antigens? This is where memory kicks in. This is where we're going to see the benefits of memory. A primary response is your immune response that occurs the first time you're exposed to a particular antigen. All right, antigen here is abbreviated as AG. Secondary is your response the second time you get exposed to the same antigen. So let's take a look at what's going on here on this diagram. So on the y-axis over here, antibody titer, that's the concentration of the antibodies in your body fluids. And then down here we have time. All right, so the first time you get exposed to an antigen, there is a latent period where you're not producing any antibodies. This is your specific immune system. You have to be exposed to it before you make a response. So all that rigmarole we talked about how you've got to activate your T cells and then activate your B cells and start making plasma cells that are going to secrete antibodies, takes a while for all of that to happen. It can take a few days. That's called a latent period, where you've been exposed, but you're not producing any antibodies yet. Then you start making your antibodies. Okay, and so the purple line or dark blue line here, that's the sum total of all the antibodies you're making. And down here, more specifically, they're showing you in red, that's IgM class. And then in light blue, that's the IgG. So the first time you have a response, you make IgM antibodies for a while, then those plasma cells really shift and mostly just make IgG. And remember, you've got those other classes in there as well, which is why the red and the blue lines don't add up to the sum total here at the top. All right, and then let's say you've got some variable time interval here. It could be days, could be months, could be years where you haven't been exposed to that same pathogen again. But let's say you get exposed. This is your secondary exposure. Look what happens. There's really no latent period. You immediately start cranking out lots and lots of antibodies. And notice you immediately start making lots of IgG along with some IgM class of antibodies that will attach to that same antigen. So that's the benefit of this is where memory has kicked in. During your primary response, you generated those memory cells, remember, and they have been sitting around during this whole time interval here between exposures, poised and ready to see if you ever get exposed to this same antigen again. And when you do, those memory cells become rapidly activated. So they rapidly undergo clonal expansion, and you start secreting lots and lots of antibodies. Notice how you make a whole lot more antibodies the second time around than the first time. And this is going to happen so quickly, in all likelihood, you won't even have any symptoms of it. You won't even know that you're sick, because these antibodies are going to get cranked out so quickly, they're going to go around and tag the microbe, and it's going to get cleared out before it's capable of beginning to damage you. So that's the whole basis for how immune system memory works. All right, so the next video lecture, we're going to start talking about how the immunity that's generated here with your third line of defense, T cells and B cells and the memory that occurs, can be classified into 4 categories. And you may have learned this before if you've taken Biology 202. It gets covered in that class as well. We'll talk about it maybe in a little bit more detail in this course. And then that's going to lead us to an extremely important topic, vaccination. And that's what we'll talk about to finish out Chapter 13.